So good evening, dear friends and colleagues from the space of ophthalmology and oncology. Welcome to our distinguished ophthalmology lecture series. Following the inaugural lecture delivered by Professor Watson back in June, we are delighted and privileged to have Professor Tony Mok to speak with us this evening. One important mission of our lecture series is to engage our academic community to advance and transform clinical cares of our patients. Designing and performing clinical trials is no easy task, not to mention completing clinical trials that reset the paradigm of care. Professor Tony Mok is a quintessence model. May I now invite Professor Vivian Lin, our executive associate dean and interim head of the Department of Clinical Oncology, to give a welcome speech. Professor Lin.
Thank you very much, Chris. And as executive associate dean, can I warmly welcome everyone to this evening's lecture, the second of the Hong Kong U Ophthalmology Distinguished Lecture Series. I'd really like to congratulate the department for this series. I think it's a fabulous opportunity to bring people together, whether it's about community ophthalmology or ophthalmology education or research, and going beyond ophthalmology as well. And tonight we see a kind of bringing together of different fields. And so, as the interim head of the clinical oncology department, I am, of course, absolutely delighted that we have such a distinguished speaker who is extraordinarily well known, not only to the oncology field, but to really all of Hong Kong. So I think the series, will be great in terms of having Dr. Lee, who has graced us with his presence this evening as well, being the inaugural speaker in the first one. I'm sure there are many more events of huge quality and very exciting to see in the future. So without further ado, let me turn it back to you to introduce our speakers, so I'm not taking any of the precious time. Congratulations and welcome to everyone.
In fact, I think every one of us here know Professor Tony Mok very well, and I don't think I would spend another minute. I would just invite Professor Mok to come to the stage, and then following the talk, we'll have the Q&A session. Professor Mok, please.
Thank you, Professor Lin, and thank you to Christopher, my good friend from CUHK in OLEX, for inviting me to this distinguished lecture. This is my disclosure, and this is the free company that I'm on the board of director. I was delighted to come to speak, but I had a problem. What is the similarity between ophthalmology and oncology? What is the topic I'm going to speak on that will be interesting to both parties? So I think, and all I can find is an O, and there's logic. Apart from that, there's really not much in common. So, if in doubt, I have to resort to one thing I like the most, and then everybody will share common interest, is food. So, first question, what is this dish? This? What does it look like? Anybody? French onion soup. Not quite. It's actually a Chinese dish, designed in a Western style. All right, I will just let you know now. It's actually sundak. So it is actually made of the gut from the fish, together with a bit of fish liver, baked in egg and fancy with the top at the double. Taste the dish. However, there's one problem with this dish, is that they just cannot get rid of the fishy smell, the sangry. So one day I was invited to my friend's home for dinner. He's a retired lawyer, very, specialized himself in the fine cooking at home. So he presented this dish. So it looked like gochujang, it smelled like gochujang, it tastes like gochujang, but there's no sangry, no fishy smell. I said, what is going on? So brother eventually told me, instead of fish gut, he used French oyster. Instead of fish liver, he used foie gras, goose liver. And then made it the same way, and you got the texture, and the taste, and the smell, but without the sangry. This is transformative. So, the art of the transformative cuisine, you can keep the idea of creating one, the recipe of doing how to do it, and finally the execution of doing it. But on the other hand, a transformative clinical trial is exactly the same. The idea, the recipe, and the execution. And for that, it's translated to the study concept, the design and protocol, last but not least, your study team. So tonight, those are the three areas I'm going to share with you my personal experience on the idea, the recipe, and the execution. Starting with the idea. Now, clinical trial idea, where does it come from? First of all, any transformative clinical trial has to be coming from a transformative question. So everything starts off with asking the right question. Usually, most of the time, in any specialty, whether oncology or other, is asking the question, is A better than B? But this day, that may not be good enough. We have asked, is A better than B in a selected population? So first, focusing on is A better than B, I'm going to share with you one clinical trial in oncology. Sorry that I really don't know much about ophthalmology trial I can quote, so that's why I find this one. So this is ramucirumab, which is an anti-angiogenic drug, together with a chemotherapy, docetaxel, compared with docetaxel in a late-stage lung cancer. You can see it is a positive randomized phase 3 study. Ed Garon, my good friend, published in Lancet Oncology, in Lancet in 2014. Not bad, a Lancet paper. Now, it is slight improvement. The median is 4.5 months versus 3 months. Little bit improvement. Overall survival is 10.5 months over 9 months. And you'll say, what the heck? How much does it help the patient? But let me just dissect through what had happened. So first of all, they identified the patient with advanced stage lung cancer, late stage, treatment A versus treatment B, and then you find a primary endpoint. That is the usual study design for most of every specialty. It can be cancer, can be an eye problem, can be a heart problem. That's the usual thing. But in the design, first of all, the selection group, you have to assume they're homogeneous. So every patient has to be the same. That's why you randomize them. The most you can add some stratification factor to make sure some key factor are balanced between the two. But are they really homogenized population? Treatment A, before you start the study, you have to know, is it really that good a drug that make a big difference? And finally, you choose an endpoint that will make a relevance to the patient. In this case here, that's the survival. But then, this is a positive study. How do they make it positive? So because they actually doesn't think that it makes such a big difference, what they have done is assume a small difference and then getting a very huge sample size. This is the study design. They used 1,200 patients to look for a very small difference in hazard ratio of 0.816. So in the beginning, they don't even trust their own drug. They just want a positive study. So as a result, it's a positive study, it's a Lancet article, but it really doesn't help the patient much. It just, this drug is only mostly used in the United States, but outside the United States, very few countries will adopt this drug. So yes, it is a clinical trial. It's a positive, it's a Lancet trial. However, is it a transformative trial? In my opinion, not quite. Actually, I'm the one who wrote an editorial on this study. So now we move on. To cancer, we start to ask the question, is A better than B in a selected population? And again, it's not just cancer, cardiology, diabetic patient, ophthalmology situation, all more and more, we're talking about selected population. But once we have a selected population, you have to talk about the target. I like this picture because you think you can select population and hit the target. If you hit the target will fall. But you hit the poor guy holding the target will also fall. So as a result, even you see a target fall, you may or may not know whether you have hit the target properly or not. So what you have done, that is to identify a driver oncogene in cancer, but in our situation, you have to find a biological target that is meaningful to the patient. Second is to use a laboratory to define the biomarker, and lastly to prove that it worked in a specific group or selected by biomarker. So I'm just jumping right into the biomarker. What exactly do we mean by biomarker? Now, we always thought that we have a homogeneous population. On the superficially, when you have lung cancer, you thought they're homogeneous. Or superficially, you have one disease, you thought homogeneous, but actually there's a composition of multiple different genomic situation or other pathologic situation. We can't group it together because it's for convenience. So among which, you got your green guy, your yellow guy, your red guy. But if you have a biomarker that can select all 5 green guy out of the 5 green and out, you have 100% sensitivity and 100% specificity, and that is a great biomarker. But this doesn't exist. Okay, there's never had a biomarker that is so good. But then it's a not so good one is that you can take 4 out of 5, and then you also include some that you thought they're green guy, but they're actually yellow or red guy. So as a result, you got some false positive and false negative. That's a reality situation. So the first rule of the thumb is that you have to know how good your biomarker is, how accurate it is before you go into clinical trial. But to understand the biomarker, you have to know whether they are qualitative, which is yes or no, absolute person, or whether it is quantitative, is by measurement, like the EBV DNA in nasopharyngeal carcinoma. It's a number of copies that you can detect in the blood. So it's a continuum, and then for that, you have to decide where's the cutoff. So biomarker, first thing, is it qualitative or whether it's quantitative? Is it qualitative? Whether it's objectively measured. This applies to, again, all walk of medicine. Now, so, and then you thought that you had the biomarker, you thought you had a cutoff for the qualitative biomarker, you have to prove it. And then you need a validation process and standardization method. Now, some of the standardization method is easy, some are more difficult. If you look for a gene, it's easy to standardize it. But if you look at protein expression, it is how much the pathologist is looking at it. So in the eye of the beholder, whether it's 2+ or 3+, it's variable from person to person, from pathologist to pathologist. So it's more challenging. And after that, you still need to do your validation trial, whether it's retrospectively looking at it. A lot of the so-called young postdocs will love that because all you need to do is go back to the file and then take out information and do it. But then there is a lot of limitation due to the retrospective nature of the study. But better, you do a single arm trial to see whether it's predicting outcome, but you cannot confirm that until you do a prospective randomized study. And here it is that usually when you go to phase 3, that you try to integrate your biomarker in your phase 3 study. So that's a usual method of approach to a transformative phase 3 study, is to understand your biomarker, know how to quantify it properly, how to validate it properly, and then integrate it into your randomized study. And then the efficacy. Now the efficacy, there's actually only usually 2 ways of designing the study. One is called a marker of treatment interactions design, is that you include everybody, and then you do a biomarker, one group that's positive, you randomize it to drug A versus control, or is the biomarker negative, you randomize it to drug A versus control. And then you do interaction test. This is a perfect study. However, it's very expensive. Most of the people doing is a market-based strategy design, is that you only randomize the positive group, and then you see whether there's a result. And usually that's still acceptable, although it's not that perfect. So those are the study design. Now, there's a whole bunch of theories and it's kind of boring. So I'm going to go back a little bit to share my experience on how we can move, from asking a simple question to changing the paradigm. So asking a question is an art. You can learn that from a lot of interviewers, but poking into the right question, if you can get the answer, and then in a surprising manner, then you get a lot of information. But usually, when you do clinical trial, that you have two things that's going on. The first question is what is available versus what is relevant. Sometimes you look at what is available before you ask the relevant question. This is a Chinese called 太松食法. So if you are in the ophthalmology department, you are not going to do a clinical trial on this melanoma of the retina because you're not going to get the sample size. It is not available for you to ask a relevant question in Hong Kong due to the limitation of the sample size. But then there's another way, is to ask what is relevant before you ask what's available. So in other words, whether you can create the availability. Now that is actually usually a bit more transformative. Because it's all the time, you just 太松食法, then you're confined by your limitation. Hong Kong is a small place. If you always look at the confinement of the Hong Kong and without looking outside Hong Kong, then your chance of having a transformative study is actually less. So I usually like to ask the question, what is relevant, before I ask the question, what is available? And if not available, then I ask, how can I make it more available, or at least a little bit more available? So the direction of thinking of asking the question. And then another rule about asking question is that don't make any assumption. A lot of time that in clinical trial, we have a lot of assumptions that say that, you have to do this way. But under the assumption, again, you limit your relevance. And think a bit deeper, and then always say, no question is ever stupid. A lot of good thought comes from stupid questions. So challenging orthodox is good, but challenging orthodox only in a rational manner. As long as you were able to make an argument to challenge orthodox is acceptable. Now, I'm just going to share with you the story that the EGFR mutation. Now, EGFR mutation had been around for a long time. Stanley Cohen in 1986 received the Nobel Prize for discovery of the epidermal growth factor. This is a factor on the cell surface that helps to signal the so-called the growth of the cell. And they have been trying to target over the next 20 years how to control the cell growth by targeting the EGF. And as you can see that there is multiple channels. One channel is called the RAS pathway. The other one is the PI3K pathway. And both ways is go down to the nucleus and cause the cell to duplicate. So if you're able to block the receptor level with a monoclonal antibody, you may stop the signal. Or you may just block in the intracellular level, in the tyrosine kinase, you may also block the signal. So there have been drug development. This is the first generation antibody for that, and there's also the first generation EGFR TKI inhibitor. So those drugs have been around since the 90s. So they do clinical trial. These are the two first clinical trials called the IDEAL-1 and IDEAL-2. And you can see that IDEAL-1 was done in Japan mostly. The response rate is only about 20%. In the United States, only 10%. The progression-free survival is 3 months in Japan and 2 months in the United States. What happened? I mean, how is this going to transform the lung cancer care? However, it was quoted in Forbes magazine as the miracle pill. Why is that? Because there are occasion patients who got dramatic improvement. The cancer did basically melt away. Not many, but some patients had melt away. So much so that actually they go to the so-called FDA and request for this drug for a special release at that time, back in 2006. And then Roche did a study trying to randomize against a placebo control in a late-stage situation, Response rate, again, 9%, median progression 2 months, over survival 6.7 months. Again, very disappointing. But then the drug was approved because there's actually no better choice at that time for patients with late-stage lung cancer. Now, and then we have to ask the question, now, can we get this drug into the first line as a standard therapy? But come on, less than 10% response, 10% response rate, chemotherapy is actually better than it. The first question is that we cannot assume it's better, and how can we make it better? Another obstacle at that time is that in United States FDA, all the study that's approved it has to use overall survival as the primary endpoint, meaning that how long the patient survived rather than progression-free survival. So I was given the task to how to design this study, and the 2 questions I have is, number 1, how to select, and number 2, can we change the endpoint? Now, the reason of how to select is this, is that by that time, TGF mutation was not quite discovered yet. And it's only 2004 when TGF mutation was selected, what was discovered in Harvard. So, but all the time, we know that something is cooking in terms of the biomarker selection. And then the next question is at the endpoint. Because anybody, if you have this mutation, if you do not take it in the first line, and you take it in the second line, you see benefit. So once you get the crossover, your so-called addition to the survival is limited. So you will not have a positive study with over survival. So with this discovery of the mutation, a single point mutation, exon 19, deletion in exon 19, and a point mutation, exon 21, this changed the whole paradigm. So at that time, I still remember that I was in the audience when this was presented, and I told myself, because that is actually the one thing that I've always been looking for, the marker to select the patient. So as a result, we managed to publish this paper, Transformative versus the textual carbo in the lung cancer in 2009. And then what we have done is that able to recruit over 1,200 patients, demonstrate that if you're mutation positive, if you're giving Transformative, you are actually better than chemotherapy with hazard ratio 0.45. And then if you are actually mutation negative, the chemotherapy is better than the hazard ratio 2.85. And the interaction test is less than 0.001. So this is actually with the design that I just told you, the microstrategy design. And this is the one study that established personalized medicine for non-small cell lung cancer using EGFR mutation. So asking the basic question of selection and changing the primary endpoint to progression-free survival instead of survival, we were able to transform the care of lung cancer. Now, that is one experience. The next question is that, how do we get there? What is the recipe for knowing the mutation or knowing the concept and then moving it into a clinical trial that was started in 2005, completed in 2008, and published in 2009. So what is the recipe behind this? How did we do it? Well, 2 areas I'm going to touch on. Of course, there's a lot more. First is the stakeholder. Second is the protocol. Now, stakeholder, usually in most of the drug trial, or actually instrument trial included, there's academic and there's industry. We always see industry as the dark side because they make money. But the fact of the matter is, yes, they intentionally make money, but we cannot serve the patient well without the industry support, because that is exactly how the world works. University have to create company before they can distribute the product. But it's a matter of process how. But then, if you look into the objective, we like to address the clinical question. Industry, because they got financial pressure, they need a drug approval. So, they're not so separated. We have to find our intersection. Now, the intersection is to say, what is the reason behind a clinical trial? Now, for us, scientific interest is number 1. Followed by publication, of course, the best is international, big journal. And then with that, you actually end up with some gain, which is academic promotion. I'm sure Professor Lim looked at your CV seriously to see how many papers you can one by one. And very little is a financial incentive if you are stopped into the item. So the importance is from scientific interest and go downward. From the industry, drug registration from US #1, Europe is other, and then China, Japan #3. Followed by scientific interest, and then the market value or the indication, and then what indication. So their importance is go this way. The question is that, how do we match them? Because we have some shared entity, priority is different. So it doesn't mean that one is exclusively not for the other. So the way I see it is that we want to optimize care, they want to gain drug approval, this is coming soon. Okay, so how to link this relationship is put patient benefit in the center. Use that as the key argument or common point. Industry has to provide patient care before they can be financially feasible. Doctor has to provide patient care before we can justify or doing a clinical trial. So whenever you design a clinical trial, you have to benefit all three parties. Keep them in mind, keep the stakeholder in mind, including the industry, your patient, and the investigator. And you have to balance the benefit of all three parties. One point gain too much is not good. All three parties has to have an equal stake in your clinical trial before you can promote your clinical trial. I think, again, this applies to all walks of medicine. Protocol, big and boring. I hate it, especially if you have to write. This day I'm good, I can have the people to write for me. But how do you mitigate through a thick protocol? Biomarker talk about, I'm not going to talk too much about it, and start right into the intervention. These are the 4 major areas that you will focus on. There's a lot of quote-unquote variation, I have to say, alternative pace, but then this is the 4 areas that you cannot miss. When you go through a big protocol, those are the 4 things that I hunt for. Intervention, or biomarker, intervention, endpoint, and bias. Biomarker, I talked about, now the intervention. So the intervention, the first thing I ask is it biologically sound? All treatment comes from biology. If the biology that you understand doesn't make sense, it won't work. So you must have a very good biologic basis. And then the next thing you do that is to look into the early data. And you have to have the ability to evaluate how true those preclinical data is, including the preclinical and animal models. And then the toxicity profile has to be clean. And that is actually related to the basic biology of the drug as well. And last but not the least is the routine, the route and the schedule of administration, in other words, the scheduling and the pharmacokinetic. So all those are basic things to understand about the intervention, but then don't trust the drug company at this point. Okay? I said I work with them, that doesn't mean that you have to trust them. I was going to say that you can't trust your girlfriend, that you don't always trust her. But you have to be critical sometimes of what they tell you, because they always tell you the good stuff. They give you good news. They give you the strong information. They hide the weak information. So you have to be very critical at this stage. Otherwise, once you bought into the concept, you buy, you sell, you sold to it without being critical, then you may go on the wrong path. So be very critical of the early data, especially the preclinical data. And then, when you do a randomized study, it's not just the winner. You have to see the potential loser. How bad is the potential loser? It is a weak guy, the chance of winning is better. There's a strong control arm, the chance of winning is actually less. So you have to understand what is the FDA current approval with that. If the drug is actually very effective, you're in trouble. You have to find a drug actually that's not too effective, but still standard. Okay? And then it's actually also have to be able to identify what is the weakness of this particular control arm. Because not all drugs are the same. So, for example, in cancer, not all drugs can penetrate the brain. If the one drug that you know that doesn't penetrate the brain well, and then your study drug can penetrate the brain, you specifically identify brain metastases and also put an emphasis on it in the management. That's exactly what I did with the drug called anatomy, which is another New paper that's compared to the standard of softening. So again, knowing the weakness of your control arm. to the endpoints. Endpoint is important. Why is it? Well, first of all, that is represent your hypothesis. And second, is the fact that it's a foundation for sample size calculation. Sample size equal to time and money. If you calculate it well, you calculate effectively, that gives you a much better chance of having a successful trial. And last but not the least, when you write a manuscript, your endpoint will be the first sentence in your conclusion. I actually always teach my students is that if you want to write a manuscript, you start with your protocol. If you want to write a protocol, you start with the manuscript at the bottom line. What are you going to say in the conclusion of your final manuscript? That is the starting point of your protocol. So in a way, if you want to conclude that this drug is better than this drug in these circumstances, that is your hypothesis, and that's your endpoint. And how? So this is actually an important part of the whole development. So, but then there's deep philosophy behind how do you choose your endpoint. Now, it is philosophical. Go back to this workout study. So it is a drug approval, it's a Lancet publication, it's minimal benefit. It's not the one that you want. So you select among the various endpoints what is the optimal for your circumstances. And then in ophthalmology, I try to read up what is good and relevant. This is all I can find. Visual acuity. Best corrected visual acuity, patient-reported outcome, some standard automated parameter of a Homer or some structure in endpoint. But I actually start to worry with you is which one are they subjective and which one is objective? Because when you measure acuity, I see blur or not blur, is it so easy to get my subjective feeling of how acute my visualization is? So I don't know. I mean, I'm far from understanding ophthalmology, but when I look at this among endpoint, I would just say, how do I justify and measure very accurately? Last but not least, also they have bias, all the way from selection bias, performance bias, attrition bias, detection bias, and reporting bias. But the fact is that you can actually minimize the bias by designing a protocol properly. You have looked into all this potential bias from the very beginning. So for the sake of time, I'm not going to go into any great detail. Last but not the least is execution. Now, just like anything, you want to be a good chef, you need a good team of chefs together, seamlessly working together. So the key question now, especially for the young investigators, is how do you build a team? Now, let me just start with my story is that this is how I started as a dumb-looking kid. That I went to Canada, studied medicine, finished my medical school, and then I practiced in a place called Scarborough Grace Hospital, a small community hospital with about 400 beds. And then I was the only Chinese-speaking oncologist there, doing my humble work for 7 years. By interesting chance, I actually come back to Prince Royal Hospital up to the Chinese River, Hong Kong in 1996. That was 25 years ago. I go from a very cold Canada to very congested Hong Kong. There's a lot of adjustment. But I was 36 years old. I have 0 publication. How did I start? At age 36 with 0 publication. So I give you my lifeline. I come back with 0 publication. And then, 1998, someone come to my office say, I'm from LA. I got a new chemotherapy called Transformative, which is a very old drug now. And I had $20,000 US. Would you want to do a trial with us? $20,000 US to do a clinical trial? Now they are saying, forget it. Don't waste my time. And that time I say, yes, thank you. I was desperate. But then how do you cook a dish with $20,000, a trial of 40-some patients? I go and ask help from a lot of people. My colleague from radiology, colleague from the lab, thank you. So basically, you assemble your team to support you with the minimal so-called support that you can get at that time. So that was actually eventually published a single-arm study in cancer as my first article. But that springboard me as someone who can do clinical trial. Then I was invited by multiple countries around Asia to talk about the drug, oncology, and clinical trial. So from there, I started to build a team, and then I established a lung cancer research group, which composed of oncologists from China, Korea, Singapore, and also from Malaysia. So together, about 9 centers, we worked and start doing a bigger protocol and bigger study. And then the opportunity of IPAS combined, that was the study I just shared with you. And that is the one that gave me the first $1 million article. And from there on, I become the president of the International Association of Lung Cancer. I published a total of 40 million up to now. And then now I'm going to retire and die. But then the question is what happened in the first 10 years? Now, it's a long time, man. I mean, you may see this successful end of my career, but have you seen me the first 10 years with little resources? What did I do at that time? Building a team. It takes time, it takes effort, it takes inspiration to build a team. So if I have any advice for you tonight, is to build a team around you if you want to do clinical trial. So, but then you got a team, but how do you become a leader? So ask the next question is, what exactly is leadership? Well, those are great leaders, my favorite leader. So this one got Nobel Prize as well, but I don't know whether he deserved it, but he's still a great leader. But on the other hand, these two guys are also great leaders. Totally different style. Did something really bad to the world. But they are leaders. Why? They influence others to share the same thought and same idea. And leadership is to share the vision and bring the group to execute the vision together. So leadership is nothing but just a power to influence others. The question is that how do you build this power and ability? So to me, there's a 3 component. I call it SIM. Basically, you have to create vision for the future. You have to see something that others don't see so that you can share with it. Second, you're able to inspire and motivate people to engage in the vision. Last but not the least is to manage delivery of this vision on a day-to-day basis. So those are the few tips that I'm going to share with you, is that you will influence others, you create leadership in your team by the way you think, the way you do, the work you do, and also the words you say. Now, thinking is important. First is the forward thinking. You actually have to plan and look at the future and create a vision of your own. You have to see how, to me, is how the lung cancer should be managed in the future. What are the ideas I bring into it? So for that, you need the courage to think ahead, and you probably have a certain amount of emotion that you have to have some EQ when you're being criticized. And then you have to have the strategy of planning from it and commit to it from the bottom to the end. Critical thinking is also very important because you were bombarded with information. If you're being critical, you're able to understand what is the right information for you or for the situation and what is the wrong signal. Especially this day, we're overrun with so many signals from all the internet information. You have to be critical. So interpretation and analysis is the most important. Now for the young doctor, I would just suggest that you have to be objective and logical all the time. And then practice your critical thinking in various walks of life, and especially if you want to do journal critique. I know it's a lot of work, and you don't get paid, and you don't even get recognized sometimes. But doing journal review is actually giving you the best method to be able to think critically at your own specialty. Because you are criticizing the top of the information that's being given, and they're accurate information that you just have to be able to see how good are those information. And then the work and work. For time, I just condense in the version. Is again about publication. Now, publication starts with the concept, but then you don't always get to publish your concept. So you really have to seize the opportunity the best you can. But then, the more you get the concept and you get opportunity to publish, the opportunity will bring you more concept. So it is a vicious cycle. So you always look for this opportunity, But every time when you have a concept idea and publication, ask whether it's new, actually whether it's going to change practice, and also whether what's the strongest scientific message you can deliver in the publication. And then there is also another important aspect for young doctors to participate in something called advisory board, because that is where the industry seeks your opinion. A lot of the time we may be reluctant to participate, but actually this is an important step, especially the international advisory board. A lot of the advisory board, Oncology advisory board, is more marketing stuff. Those forget about it. But then if you have an opportunity, go to the international advisory board, because you will be needed there. Your opinion will actually change the industry, and then there is a place where they identify the key opinion leader. If you never appear at advisory board, there's a very little chance that you will lead an international study. And then, when you're there, you cannot just sit there. You have to be there, be very vocal, and know who is who's making a decision, and provide a generous opinion of your own and share with them. So that is one place where I call the fighting ground for the young doctor to become an international leader, is actually in the advisory board. And last but not the least is to become a study PI. You actually have multiple ability. It's not just to do trial. You have to analyze the data, able to present the data appropriately, able to write a manuscript, and eventually you have to trust the data and become a spokesman for the data. So it is a long journey. It is an interesting journey, and it takes a lot of you to become a leader in the clinical trial team. So in summary, I think the idea is the fact that you have to ask a relevant question. You have to learn how to select the population, and in my view, it's the biomarker. The recipe is that you really have to develop a relationship with the stakeholder, a balanced one, including the patient, the industry, and the investigator. And also in the development of protocol, you need to look at the intervention endpoint advices. Last but not least, when you execute a trial, you do need to build a strong team, and then you have to become a leader, and you have to criticize your own thinking, and you have to modify and refine your work and your word that you say. So under the note, I thank you all for the opportunity to share this experience of conduct transformative clinical trial. But there's one transformation that I really have to share with you. This is actually the Hong Kong College of Medicine in 1908. And now you guys are ranked as number 34 of the world, the best medical school. Congratulations, and thank you again for the opportunity. Thank you.
So, thank you very much, Professor Mok. A lot of insights and wisdom in the very talk. I'm sure we all learned a lot, hard skills, soft skills that you mentioned. Now, let's actually, this is a Q&A section. I'm sure you have time for Q&A, but let me have the privilege to ask you a question first. Now, talking about endpoints, you emphasize about sample size calculations and endpoint selection. I think one challenge that we have in conceiving clinical trials in terms of sample size required is that it's not easy to get it right, and statistics can be a tricky business. Now, I did some homework. I studied some clinical trials that were done in old days and some of the more recent clinical trials. And I noticed that very often when you apply a test, comparable sample size, say for example, because proportional Hessian models is the most popular statistical tool used in sample size calculation, but very often we don't see people, when they did their sample size calculation, take into consideration of covariates like the patient performance status, like the biomarker portfolio, and it's a rather simplistic kind of estimation of how many subjects would be required for study. Now, I would like to know, learn from you about how to actually get the sample size done right. given the fact that we face so many variables, so many complexities in the design of clinical trial?
Simple answer, don't do it yourself. Basically, we're doctors. We may learn and understand some of the statistics. We're not expert. Just like I would not try to treat someone with an eye problem, and I hope you do not treat someone with lung cancer either. So there are experts that you can sit down at the meeting with and discuss the various aspects. A good biostatistician will have a reasonable understanding of your clinical situation. And for us, we are actually clinicians with a reasonable understanding of the biostatistic, but we're not the expert on it. So if I try to use that as my own way of thinking, I'm not objective enough, and I'm not using the most expert skill of mine in the situation. So it does take a team effort, and a good biostatistician is definitely part of the team. So that's number one. Number two, that is actually an important part about the biomarker selection, because they actually try to minimize your confounding factors. Because most of the so-called, at least in cancer, is a genetic, so-called the driven disease. There are certain similarity among cancer. So by a biomarker selection, you already narrow down your candidate, and that will actually minimize the risk as well. So that's my 2 points. Unfortunately, I did not give you a right answer, and then I really do not pretend that I know too much statistics. Right.
It's important for message, always a team of good statisticians, particularly so that you can always give advice from them to get the sample size calculation right. And it's actually tied to also endpoint selection, which you also mentioned. 2 common endpoints that we see in the cancer type research is overall survival, and we have also progression-free survival. And so which one generally would you use? Because I would guess, like, we talk about also endpoints in ophthalmology trials with visual acuity, we have also other endpoints. We don't use, say, for example, visual acuity very often as an endpoint in glaucoma trials, because it's actually quite a lazy event. I think similar analogy we have here, overall survival could be perhaps a less sensitive endpoint, I believe, but it's actually easier to measure. But progression-free survival is actually quite challenging to measure. So how we select endpoints?
Another important question. So first of all, the endpoint has to be maintained not by you, but by FDA. So you have to play the game of understanding what the FDA may require on a specific indication. Now in cancer, the problem is that after the first line treatment, there are multiple effective line treatment available afterward. So in other words, if you put in a variable in the beginning that you cannot deliver towards the end, you are in trouble. So what happens is that if you have first line trial of a drug A, but then you have no idea what may happen to the patient afterward. If you over trial, you are limited to the random of what the final outcome is. So the best is to control what happened during the trial, which is progression-free. So for the first line, meaning that the first treatment the patient received, I would definitely fight for progression-free survival. The end of the line treatment, then you may consider using the overall survival. So that is the usual paradigm. As a matter of fact, FDA, before the IPAS study, they always use overall survival. That is actually the first trial ever in cancer to use progression-free survival as an endpoint. But since then, they have accepted that. And number 2, progression-free survival, it may sound difficult, but it's just a matter of investment. Meaning that how frequent you monitor the either tumor or the endpoint. So, like in a randomized study situation, that we do it frequent enough to look at the tumor, then we actually can measure the progression-free survival very nicely. And not just that, we are actually, for example, we sometimes use the brain, as an endpoint, and then we actually do MRI of the brain every 6 months. So it's costly, but actually that is actually crucial. If not explained to the industry, then they actually would take it as well. So it's a matter of the circumstances and the FDA preference eventually.
Now, talking about design of clinical trials, we know that recently people beginning to talk about, because clinical trials are expensive, and we were talking about real-world patient data. So what would you see future of clinical trials like in various clinical space and
Right. So for the common disease, I personally do not see the real-world data to replace clinical trial in the registration process, because there's no justification to do that for common disease. For the uncommon disease, potentially I think it's feasible. So for registration, that would be my take. But then if you look into real-world data versus the randomized data, then a lot of this is actually the confounding factor at the endpoint. If you actually have the real-world data from, let me just take extreme, from Eastern Europe, patient may have one eye treatment and no more, survival is shorter than, well actually that even applies to UK, unfortunately. But then if you come to a country like United States, and they have multiple line treatment, and then better support care, then their living is longer. So the real world data is actually variable geographically and also according to the system, the healthcare system. So how much you can take into this variable is actually debatable.
So, two questions on the soft skill. You share with us tremendous insights in how to navigate among the industry and the other study sites, investigators, patients, and it is remarkable that some studies, that some clinical trials that have done involved over 200 study sites. So how would you actually coordinate such a large study? How do you build a team to overlook such an extensive array of different parties and patients groups?
So first of all, when you part of industry, you don't do all this by yourself. You work with them very closely. But there's a couple of easy remarks. First, be a good salesman. You really have to sell to your colleague that this is a worthy study, and so that they are also engaged in it. Number two, be generous. Don't always just say that I will be the first offer and then you will never be one. And then you really have to share so that the publication benefit with all your colleagues. I can remember one time with my very good friend, Pasiani from Harvard, and he and I co-lead a study. And then we were going to submit to Lancet Oncology, which actually got accepted. And then it's over the phone that together with industry and other partners to decide which one of us is going to be the first offer. And we're a good friend. I say, why do we toss the coin? So I went ahead, you tail, and he won. So he became the first offer. So, be generous, in terms of your so-called publication rights. And then with that reputation, people will work with you closely. If you are known to be the one that I always want to talk and I always kind of say you work for me, no one will want to work with you eventually.
It is remarkable. And so you talk about like death and retirement, and just want to know, I mean, you've done so many masterpieces, and then when you look back, which piece of work would you consider to be your legacy?
Well, I think the one that I share with you, that is still considered to be a legacy in the lung cancer field. As a matter of fact, the citation is now close to 9,000. And then, a lot of so-called the molecular target therapy randomized study is still model after this type of trial. So, in a way, I'm lucky to step into this position in the right time. Just imagine that if I did not build a lung cancer research group in Asia, just imagine that they did not discover EGFR mutation, just imagine that the EGFR mutation is not more common in Asia, I would never be able to do that trial. So you can say, yes, there's some hard work, but at the end of the day, there's also some luck. So I also advise you as an investigator with telephone survey, maybe that will bring you some good luck.
Right, so perhaps I think it is time for the audience, if you have any questions, and I think it's a good time for you to ask.
Professor Mok, great talk. many of you don't know me, but I have lung radiation oncologist, know Tony for 15 years actually, before you become president. So his figure in the World Lung Cancer Congress cannot be higher. He is my favorite president of World Lung Cancer, many of them, but I think Hong Kong is small, but Professor Mok in the world in lung cancer is huge. It's very huge. So what I would like to know, I think it's also important for our young people. How can Hong Kong is small? How can we make Professor Mok in the World Lung Cancer Congress so big? Not only because you don't wear a tie, because Hong Kong is too hot. So, but from your career, actually, I knew a little bit about you, first of course, from zero publication to within less than 10 years, become president of the World Lung Cancer Congress. And the organization grew the fastest among all the medical associations I have worked with, I think. So you teach us about the trick of cooking. Life is like cooking, exactly. Clinical trial is like cooking. So teach us, teach everybody. I guess we all want to know, how can you fly up so fast?
Well, I really had to attribute this to my partnership with China. China was actually not developed in lung cancer at all. But I started going back to China. I come back to Hong Kong in 1996. I started going to China in 1998. And the first city I ever visited was Xiamen. And then when I landed, there's only one airplane in the whole airport. And that's how backward those times were. But then over the years that I built very strong bonding and relationship, and all of them, the lung cancer investigators are still a very good friend of mine. And they are now world top notch. So we grow together. So I would just say that two messages. One is that don't be afraid to go to China, because they actually get a wealth of patient resources and other resources to help you research. Number two, you have to forge a long-term relationship. It's not just a give and take relationship. It's really the friendship that actually I cherish the most when I go to China and do an investigation together with them.
Thank you. Could I have one more question? The Asian Lung Cancer Study Group is still active right now? Are you recruiting new members?
No. The Lung Cancer Research Group had its mission, and actually I stopped it about 6 years ago. However, I actually have to found another group in Singapore. It's called ATOC, Asian Thoracic Oncology Group. Why do I do that? One is because there's a young investigator called Daniel Tam that I'm mentoring. I just thought that I can help him to so-called put Asia together, and he's a very brilliant young investigator. So I helped him to establish a one study group like what I did with myself in Hong Kong. So now there is Asian Thoracic Oncology Group. And yes, that group is recruiting, and that one is active.
Okay, thank you.
Any other questions?
Thank you for the talk. Actually, I was one of the students back in medical days. Thank you very much for a good talk. And I just wonder, we all want to be a research team, and can you share your experience when you are establishing the lung cancer research group? Actually, you were just around 10 years maybe in your career, so maybe at that time you may still not having a lot of strong publications, not in the New England Journal of Medicine yet. So how can you let or draw people to trust you to form that strong bonding, except using your personal charisma that you actually don't have it? Thank you.
Thank you. It is a very good question. Well, there's two aspects I can share with you. First, a mentor can actually help you. So this is actually what your mentor is supposed to do. Are you ophthalmology now? Yeah. So ask your boss. So he is supposed to help you to build your team. So there are resources he can provide, like in a way of hiring a nurse to help you. He can bring you to international conference and introduce you to the other investigation. He can also introduce you to the society at the international level, so you belong. And like, for example, I do bring my so-called young colleague, and then I also bring trial to the young colleague. So mentor got a job to do that. So you can actually look into the eyes that you only because I work for you, you have to bring me the resources to have to build the team. So number one. Number two is actually a lot of this is actually personal relationship, because you'll be able to communicate with people, become friends. That's no hard skill. You just really have to do it from the bottom of the heart. And one slight trick, everybody loves food. So if you're good at food, people will come to you. Okay, you don't have to be charming, but if you know your food, you can talk about food, then many people will come to you as a friend and say, oh, let's go and eat together. And that's how you do your friendship. Or drinking as well.
Very great tips. Thank you, Professor Mok.
So if we don't have any more questions, let's give Mr. Mok another round of applause. And it's very enjoyable. It's very candid, humble, and with a lot of ingredients. And now invite Rosaline on stage and sing private appreciation to Mr. Mok.
Thank you very much. I want your restaurant list. 3, 2, 1.
Thank you.
Thank you.
Thank you so much. Thank you.
We'll pass your left as well.
3, 2, 1.
Thank you. Thank you. Maybe we can take a good photo. Just sit here, right?